In the name of Allah, the most gracious and the most merciful. Assalamu alaikum, dear student. I hope you all will be fine. Today is 27th April 2020 and the day is Monday. This lecture is for grade 9 and the topic is electron microscope of subject biology. Students, I want you to open your book and go to page 67. Students, I hope you will be on page 67. In the front of you there would be a topic, electron microscope. So this is our topic for today and we will learn about electron microscope. If you remember the previous lecture, in previous lecture we talked about light microscope, how with the help of light microscope we can see small thing like cell, which was fundamental for all living organism. But there are some limitation to light microscope, and that's why we need to study electron microscope, or we need something which is powerful than light microscope, which is electron microscope, and that's why we are studying this electron microscope. The limitation of light microscope is that light microscope, it can show you the shape of organism, but it cannot show you the internal structure of organism. Means to say that you can see a cell with it, but you cannot see what is inside in that cell. So that's why we need electron microscope. So students, look to the first paragraph of this topic and let's see what it says. In an electron microscope, a beam of electrons is used. Magnetic lenses focus the electron beam on a screen or photographic film and make much enlarged image. Its resolving power is much better than light microscope. It can distinguish objects as small as 0.2 nanometer. 1 nanometer is equal to 1 million. Students, in comparison to light microscope, there are some differences in electron microscope, and the first difference is that instead of light in the light microscope, electron microscope are using beam of electron. They are using the beam of electron. Okay, in the help of beam of electron, they are showing you, or they are magnifying that object. And the second is that instead of optical lenses in electron microscope, there are magnetic lenses. Okay, and that magnetic lenses, they are focusing the electron. It means that they are directing the electron to object, and then from that object there is a screen, and in that screen a large image is produced. Okay, we will learn that in the next diagram. Okay, but just for the moment, it is these are the two main importance you should remember. Okay, now the resolving power of electron microscope is much greater than light microscope. Okay, it can, and it because it can distinguish object where the distance between their component is 0.2 nanometer. Okay, and 1 nanometer is equal to 1 million, mean 10 lakh. Okay, now let's jump toward the difference between light and electron microscope with the help of this diagram. First of all, we will look to the light microscope. What was in the light microscope? Okay, in light microscope is here. Okay, this is the light microscope. If you look to light microscope, the source here was light. Okay, and this light, when it was hit on the mirror, it was reflecting and it was passing through the specimen. On here on a slide we had here a specimen. Okay, and after passing through it, the objective lenses were magnifying it. Okay, here the objective lenses were magnifying it, and then another lens they were also magnifying it, and that's how a picture was created here, and the person should see that image. Okay, but if you look to electron microscope, which is on the right side, here is electron microscope. In electron microscope, instead of light there is electron. Okay, electron are using here. Okay, and instead of optical lenses, here they are using magnetic lenses. Okay, optical lenses are made from glasses, and these lenses are made from magnet. Okay, here is the specimen, mean here is the stage where you will put your sample here. Okay, and then these magnetic lenses, they are directing these electron toward the sample. For example, these electron are going in every direction. Okay, they are going in every direction, but once it's reached to this magnetic lenses, they are directing these, reflecting these electron toward the sample. Once it passed the sample, it is here on this screen, the image is produced and the person could see it here. Okay, so this is the main difference between light and electron microscope. Now let's move forward and read about the different type of electron microscope. In the second paragraph, student, if you look to your book, there is what it says. They say that there are two types of electron microscope. The first one is transmission electron microscope, which is also known as TEM. It is used to view the internal structure. It can magnify object about 250,000 times. The second is scanning electron microscope, which is also known as SEM. It is used to study the detail of surface of cell or any object. The surfaces are coated with metal. When electron beam hits the metal, it is reflected and make the enlarged image. So in the transmission electron microscope and in the scanning electron microscope, there is a prominent difference. Okay, and that is that electron microscope will make the image of those structure which is inside the cell, internal of the cell. Okay, and scanning electron microscope, it will make the picture of the surface of the cell. What is on the surface of the cell, it will just only make that image. Okay, and transmission electron microscope, it can increase the image size of a small object up to 250,000 times. Okay, and in the scanning electron, where you put the sample, beneath the sample there is metal, and these metal, they are reflecting the electron, and that's how an image is created. Let's look to it in a diagram. Now this is the diagram of electron microscope. In our daily life, if you want to see how transmission electron microscope look like, it look like this. Okay, and where are those magnetic lenses, and where are those electron, and where we would put the sample? It is here. The whole process happened in this chamber, and if you want to look inside to this chamber, it look like this. Okay, so if you look to this, here is the source of electron. Okay, here electron is producing, and these are the magnetic lenses. The magnetic lenses, they are pointing these electron toward the sample. Okay, and here is the specimen. Okay, and here is the other magnetic lens, which is called projection lens. Projection lens further straight down these electron toward the screen or toward the surface. Okay, it is on this surface or in this screen the image is produced, and this image is then shown on a screen like this. Okay, and that's how you will see the image of object. And how will the image look like? Well, it will look like this. Okay, the animal cell look like on TEM just like this, and the plant cell will look like this. Okay, now let's look to the scanning electron microscope. Sorry, let me explain here how this image is produced. When the electron is passing through the specimen, so in the specimen it is also made up of compound, also made up of molecule, and that molecule have atom and atom have also. So these component or these atom, they are absorbing those electron. So in the dense place where the cell is dense or the molecule are dense, they will absorb more electron, and when there is light molecule, they will absorb light electron, and that on the basis of that absorption of electron, the image will produce here. Okay, so that is how the TEM works. Now scanning electron microscope. Now a scanning electron microscope, it will if you want to if you want that how this light microscope look in our daily life, so it look like this. Okay, it look like this, and here is all the chamber is here, and all the operation happens here. Okay, so inside it is like this. Here is the electron. Okay, and here is a node. Now what does a node they are doing? It is also in the transmission electron, but they had not mentioned in that diagram, but in this diagram they have mentioned. So let's understand what a node it is for. Electron are negatively charged. Okay, so when they are coming from the source, they are coming toward every direction. Okay, they are coming toward this direction, this direction. So this hole in the anode, because anode are positively charged, so they are attracting toward to itself and then passing through that anode in that hole, and it going to this downward direction, and then these magnetic lenses, they are pointing it toward the sample. Okay, and this sample, it is metal coated, it is metal. Okay, so the scanning electron microscope, how does it work here? It reflect the electron back. Okay, it reflect the electron back, and with that reflection the image is produced. Okay, here is the detector. That detector reflect the electron. So if the place if the cell or if the shape of anything is like this narrow and this curved, so those places which are up, they are producing more electron, they are reflecting more electron, and those which are beneath or curved, they are producing less electron. So that's on the basis of this distribution, the image is produced in this detector, and basically this image is 3D. Okay, which is very good in quality, and it will show you everything which is on the surface of a cell or an object. Okay, and how will the picture look like in the with the scanning electron microscope? Well, it look like this. Okay, it is the structure of RBC, and here I have here I have searched this interesting diagram for here. It is the needle of a syringe, and when there is a blood on it and you try to see it with the scanning electron microscope, you will see the blood like this. You will see the cells of the blood. Okay, now let's see what is the main differences between a light microscope with the help of this table, the comparison between light and electron microscope. So light microscope and electron microscope. So first of all, the radiation is that light microscope use the light radiation, okay, and electron microscope they are using beam of electron. The light microscope is using the optical lenses, while the electron microscope is using magnetic lenses. Magnification is 10. The light microscope magnification is 10,000 times greater than naked eye, while the electron microscope magnification is 100 times greater than light microscope. Resolution of light microscope is 500 times of the human eye, while the electron microscope resolution is 400 times of the light microscope. Light microscope produce 2D images, and electron microscope TEM shows you 2D images, while the scanning electron microscope will show you 3D images. I hope you will have understand this topic. Okay, and the homework for this topic is that you should search about what kind of organism we can see with light microscope and what kind of organism we can see with the electron microscope. Okay, thank you. Allah Hafiz.